Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Improve Effectiveness of Cardiac Assessments and Early Phase Trials with Early Precision QT and AI-Powered Data Quality Checks. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, you just type your questions in the chat box by selecting the questions icon that is located on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you as well to submit your questions via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading provider of endpoint data solutions to the clinical trials industry, generating high-quality clinical evidence for life sciences companies. They offer comprehensive evidence generation solutions that combine medical imaging, eCOA, precision motion, cardiac, and respiratory endpoints. For more than 50 years, Clario has delivered deep scientific expertise and broad endpoint technologies to help transform lives around the world. Their endpoint data solutions have been deployed over 30,000 times to support clinical trials in more than 100 countries. Their global team of science, technology, and operational experts have supported over 70% of all FDA drug approvals since 2012. Now it's my pleasure to welcome our speakers. And first, I'd like to welcome my co-moderator for today's event, and he's Dr. Vic Patel. He is board certified in cardiology and holds a PhD in biophysics, having conducted extensive research on arrhythmia mechanisms. Dr. Patel spent the next 10 years in drug development and cardiac safety as a clinical development leader and therapy area head across all phases. At Clario, he oversees cardiology consulting and ECG Holter and core lab services and leads the biostatistics and medical writing groups. Next, I'd like to welcome Dr. Borje Darpo. He has held key roles on projects in all phases of clinical development. He has authored internal QT guidance documents for the design and conduct of clinical QT assessment. He represented European pharmaceutical industry on the ICH E14 expert working groups from 2000 to 2008, and later led the IQCSRC study, which validated the concept of applying exposure response analysis on data from early stage clinical trials to replace the TQT study. And finally, it is now my pleasure to welcome Dr. Jean-Philippe Couder. He is a scientist with extensive experience in the development of innovative technologies and quantitative electrocardiography and cardiac safety. He is internationally recognized for his contributions to computerized methods in cardiology. He is the founder and director of the Telemetric and Holter ECG Warehouse, a public-private partnership developed with the U.S. Food and Drug Administration and funded by the National Institutes of Health. He also served as a special government employee at the U.S. FDA from 2015 to 2022. His work has led to multiple translational scientific advances that have been featured by major media outlets, including the Wall Street Journal. Now, with the introductions out of the way, it is now my pleasure to pass the mic over to Dr. Patel. So, Dr. Patel, when you're ready, you may begin.
Thank you, Sonia. And again, I'd like to take the opportunity to thank everyone for attending the webinar today. This is the agenda for today's webinar. I'll start by going over cardiac safety and role in drug development, and specifically in first-in-human trials. And then that'll be followed by Dr. Darpo, who'll talk about the E14 guidance and give us an overview of how it was developed and how it contributes to the assessment of QTc prolongation in drug development. He'll also talk about our early precision QT measurement methodology. And then Dr. Kudera will talk about applications of artificial intelligence to assess ECG data quality first in human trials. That'll be followed by a wrap-up and a Q&A session. Next slide, please, Sonia. And so I also want to thank everyone for answering the poll question that we have when you registered for the webinar. And the question that we asked you at that time was, what is your organization's highest priority when you're conducting a first-in-human trial? And so here are the results of the poll question now. And you can see by far and away, the vast majority of respondents answered that data quality and reliability is the most important thing to the organization to capture from a first-in-human trial. A few more respondents said that it's important to help make portfolio decisions for asset progression, and then very few respondents said cost and time to market. But again, I think the most respondents, because if you look at portfolio decision making, you have to have good quality data and reliable data to make a proper decision about if you should progress your asset or not. So really, I think most 90% of the respondents really think that data quality and reliability is really the important thing here in a first-in-human trial. And we'll talk about that as we go on in the webinar. Let me go to the next slide, please. So again, I'll start with talking about cardiac safety and drug development, and specifically in first-in-human trials for the next few minutes. And the first thing I want to talk about is why are we focused so much on cardiac safety and drug development? Well, the reason is that every new drug entity, no matter what modality it's delivered by, it will enter the bloodstream to some extent. So therefore, it will be pumped by the heart and exposed to the myocardium, and there is the potential for off-target adverse cardiac events. So that's why we talk about cardiac safety the most when drug development. So what are these risks or adverse events that we're typically going to see in drug development? Well, the ones that you usually see with most patients. They're going to be myocardial ischemia infarction, valvular dysfunction, heart failure. You can get either hypotension or hypertension. You'll get arrhythmias, both atrial and ventricular, and ECG changes, such as QT prolongation or conduction blocks, are the ones that we'll typically see. How do we assess these drugs, these effects and risks in drug development? And again, the standard clinical assessments are the ones we use. The 12-lead electrocardiogram is the mainstay of assessing cardiac safety in all phases of drug development. And we'll also be looking at cardiac imaging using echocardiography or cardiac MRI. Blood pressure in the clinic and at home will also be used to assess changes in blood pressure for hypertension and hypotension. And then we'll use continuous ECG monitoring with patch or Holter for looking at rare events like arrhythmias or conduction blocks. And then serum biomarkers such as troponins and BNP are also valuable for modern cardiac safety. And go to the next slide. So, thank you. So what is a first-in-human study and how is it designed and conducted? Well, the first-in-human study is composed typically of two parts. There's a single ascending dose part and a multi-ascending dose part. Both of these parts will give escalating doses to patients, to participants, where there's single doses given the single ascending and repeat doses in the multi-ascending part. We're going to enroll a small number of participants at each of these phases to protect patient safety and minimize the exposure of a new drug entity where the safety profile is not yet known. And that's really the goals of what we're going to try to determine from this first-in-human study. The paramount thing that we want to know is really the safety and tolerability of the new drug asset. We're going to try to establish the pharmacokinetics of the asset, and if possible, we can even determine the maximally tolerated dose of the asset. The objective of the SAD and MAD cohorts is to really look at multiple doses in the MAD part, and this means that the MAD part can actually be a little bit longer than the SAD part. And then the SAD component will help us establish the first clinical PK of the asset so that we can use that to guide exposures and drugs doses in the MAD part to get the most information possible, best information possible. Go to the next slide. Now this slide shows a schematic of a typical first-in-human trial with the SAD and MAD components defined here in the blue and the red. And then you may also have food effect cohorts or drug-drug interaction cohorts as well in this first-in-human study design. Typically, we'll have 6 to 8 single ascending dose cohorts in a first-in-human trial, followed by 3 to 4 multi-ascending dose cohorts. Each dose cohort will enroll 6 to 10 participants, with 2 of those participants being on placebo and the others being on active treatment. In the SAD cohort, the first one is usually given as a subtherapeutic dose so that you can ensure patient safety before you go to higher exposures where you don't know how the safety will be tolerated. And then these studies have typically been done classically all in healthy volunteers, and most of the SAD cohorts are still done that way, but we're increasingly seeing that MAD cohorts, or all of the MAD part of the study, will be conducted in patients so that you get an earlier insight into patient safety and PK in the drug development process. We can go to the next slide. Now, what is the kind of information that we're really looking to gain from the first-in-human trial, and why is it going to be useful? Well, the main purpose of the first-in-human trial is really to determine if the new drug entity is able to progress to later phases in development. And what you're really looking for is safety signals, because most new drug entities fail to develop and progress further 33% of the time because of an unmitigatable safety signal. Or unfavorable PK is the other thing that stops the drug from progressing, about 15% of the time. We talk about cardiac safety because while safety is the predominant reason the drug doesn't progress, cardiac safety consists of about 90% of those events, for the reasons that we discussed a little bit before. So for this reason, it's very imperative that we have early insights into what the cardiac safety profile is, and if those cardiac safety issues are mitigatable or not. If they are, then we have to plan for monitoring and safety mitigation plans so that the drug can be used safely in later development. If these are not mitigatable safety issues, then we look to terminating and removing the drug from the portfolio. To make the right decisions about the cardiac safety profile and its ability to progress in the portfolio, data quality has to be very good and reliable. If the data is highly variable and there's outliers, this can introduce false positive safety signals, and therefore it won't give you the proper insights into the profile and safety of the drug. And so for this reason, we talk about data quality reliability is really critical from the first-in-human trial. As Dr. Kuder will talk about a little bit in the webinar, if you can gain insights into data quality earlier in the drug development process in the lower dose cohorts, and then you understand where those sources of data quality, poor data quality come from, you can correct them so that in higher dose cohorts, you can now actually get usable data that's really reliable, and you can still understand the safety profile and safety of the asset, which comes really from the higher dose cohorts. We go to the next slide, please, Sonia. And finally, I'd just like to conclude this part of the webinar. So we talked about first-in-human trials being really critical in the drug development process to get early insights into the safety and tolerability of the new drug entity, the pharmacokinetics of the new drug entity, and potentially even establishing the maximally tolerated dose. The SAD and MAD cohorts in the first-in-human trial really offer us the best opportunity for looking at the broadest range of exposures and doses during drug development, because the exposure range narrows during later phases of development. So that's why it's critical to take this opportunity to leverage the broad range of exposures. Study data quality is critical for having the best insights and data to make sure that you understand the safety profile of your asset and its probability of success. And you can only make good portfolio decisions about progressing the asset or not if you have good quality data that gives you the right insights into the safety profile and success of your asset. So with that, we can go to the next slide. And I'll hand the presentation over to Dr. Darpo.
Thank you so much, Dr. Patel, and thank you to the audience for your attention. I would like to start with giving a bit of a historical perspective on how we evaluate QT prolongation in drug development today. In the late 1990s, health authorities and of course sponsors became increasingly concerned over some cases, very few, but some cases of a life-threatening form of ventricular arrhythmia termed torsades. As it came, this rare arrhythmia was caused by two known examples, Saldane or terfenadine and cisapride. Some other drugs too, but those are the best studied drugs. Both these drugs are 3A4 substrates, and a few patients will actually have very high concentrations, and even less, so a much fewer number of patients will then develop torsades based on pronounced QTc prolongation. In fact, if you look at cisapride, we're looking at some hundreds of cases among several hundred thousands of prescribed prescriptions. So this is really a rare event. But this led Health Canada and FDA to come together and draft a concept paper on how to evaluate the potential QTc effect of a drug during drug development. So this concept paper was accepted in 2000 by ICH, the International Conference on Harmonization, a group that pulls together regulators and sponsors. So the ICH accepted this as a topic to start drafting a guidance in 2000. And we worked on this topic for 5 years, and this led to the ICH E14 guidance issued in May 2005. The initial concept paper was conservative and included elements that we don't typically do today, such as looking at human cardiomyocytes. But it also contained an important component that has become a key part of the ICH E14 guidance, the designated thorough QT study. A designated study intended for the sole purpose of evaluating potential QTc risk of a drug. Clearly later, or some 10 years later, we both on the sponsor side and on the authority side became increasingly aware of that the third study was conservative, and I'll show you why, also in its statistical analysis, and that we could achieve the same confidence in the results by using concentration QTc analysis. So that led us, by the time iCardiac/ERT/Clario, to initiate the study in collaboration with FDA and with sponsors, the so-called IQC study. That study led to the revision of the ICH 14 guidance in 2015, allowing us to use concentration QTc analysis as the primary statistical approach when evaluating a drug's potential effect on the QTc interval. And then the most recent version in 2002 allowed sponsors to also use non-clinical data to claim that a drug does not have an effect without achieving really supratherapeutic concentrations of the drug. So this graph illustrates the main differences between the initial guidance and what we then are allowed to use. The left graph shows you a typical by time point profile on the placebo-corrected change from baseline QTc in the therapy study. It's a small study with a 4-hour crossover design. The green line is the response to moxifloxacin, and the blue and the red lines are the active. And as you can see, the active drug doesn't have any type of effect on the QTc interval. When we used this analysis initially, we were asked to perform a statistical test to ensure that the upper bound of the confidence interval is below 10 milliseconds at each and every time point. Of course, statistically, this is a very conservative approach, in essence meaning that you're repeating your statistical test by the number of time points. In this case, this is not an issue because, as you can see, the upper bound is below 10 milliseconds. Why are we looking at the upper bound? We are because we would like to exclude an effect within the preset limit of confidence. So the two-sided 90% confidence interval has to be lower than 10 milliseconds. Now, if we take the same data and analyze in one statistical model, as a function of increasing plasma concentrations. This becomes a much more efficient way of looking at the data. And after 10 years of conducting the therapeutic studies, authorities and sponsors were convinced that this way of analyzing data had the same result, came to the same result as the by time point analysis, and therefore it would be much more efficient if we were allowed to use concentration QTc analysis as the primary statistical approach. Also importantly, using this approach would allow us to start using first-in-human studies to achieve the same endpoint as in a designated therapeutic study to exclude that a drug has an effect on the QTc interval. For that purpose, we designed and conducted the IQ-CSRC study. This study brought together regulators from FDA, Europe, and Japan with sponsors and academia. And we designed a study with a small number of subjects, in which 9 subjects received 2 doses of an active compound and 6 on placebo. And we tested 5 different drugs known to cause mild QTc prolongation and placebo. And then we also tested the negative compound, levocetracine. This graph shows you a typical concentration QTc analysis, a goodness of fit graph that shows the black line is the predicted QTc effect across increasing concentration levels. You can see that the black line has a positive slope, and if we look at the table below, we can see that the slope is statistically significant. We can also see that the upper bound of the confidence interval for the predicted effect in the QTc interval, the upper bound is about 10 milliseconds at relevant concentrations, in this case around 210 nanograms per ml. So this is a positive outcome using a very small sample size. More importantly, of course, we would like to demonstrate that our drug is negative. And there it's more challenging. We still need to look at the width of the confidence interval, the upper bound of the confidence interval. This is the same graph from levocetracine from the same study, looking at 9 subjects in active and 6 on placebo. So a very small sample size, but with 2 doses of active, 1 therapeutic dose and 1 really high dose. And this is the result we saw. We see a very shallow slope of the concentration QTc relationship, and we can see clearly that the upper bound of the confidence interval is below 10 milliseconds, even at concentrations that would constitute the highest achievable concentration in humans. So this is a negative QT evaluation based on a very small sample size. And of course, these results were very encouraging and led to the revision of the ICH of E14 guidance, thereby allowing us to use concentration QTc analysis, not only in the therapeutic study, but also applying the same technique in first-in-human studies. Now, why does precision for the QT interval measure matter? Well, it matters because the greater precision you have, the more narrow confidence intervals you will see of the predicted effect. So this slide shows you the main features of a standard technique and a technique we call EPQT, and which we always apply to health studies in health volunteers. With the standard technique, which we also have and which we apply in patient studies, we extract 3 ECGs per time point. And from these 3 ECGs, we extract 3 beats. We're looking at 9 beats per time point. The measurement points on these ECGs are initially placed by the underlying algorithm, and then the technician/cardiologist looks at these measurement points and makes adjustment. So this is a very robust technique. It can handle T-wave morphology changes, and we always use this technique in patient studies because it has a really reliable output result. And we have a huge amount of experience with this technique in patient studies. EPQT, on the other hand, extracts much more data at each time point. Actually, 10 replicate ECGs are extracted at each time point. All beats in these 10 replicates are looked at by the underlying algorithm and categorized into good quality or not so high, not high quality. All the high quality beads, all the good beads are accepted without any form of adjustment. All the beads not identified as high quality are looked at by the technician and then either accepted or rejected, but there is never any adjustment of fiduciary of measurement points. This means that we end up having measured around 100 beads per time point and subject with one unified method, as compared to 9 beads with a semi-automated approach with measurements also adjusted by a human. And this, of course, translates, or I shouldn't say of course, this translates into precision. This is a publication we did in collaboration with Novartis some years ago. And we're looking at the variance across time points using either a standard approach, the red lines, a semi-automated method, which is the standard method I described in the upper panel, comparing that with the precision or the variance with EPQT. We are looking at 7 different therapeutic studies. The upper panel are 5 studies with a crossover design, and the lower panel 2 studies with a parallel design. And whether we look at test drug versus placebo in the left graphs or moxifloxacin versus placebo, we can see that EPQT roughly halves the variance across time points as compared to standard technique. And this, of course, also then has a huge impact on the sample size we need to exclude a small effect, because the lower sample size you have, the more important the precision of the ECG measurement becomes. And these are simulated data that we have done internally, but also in collaboration with FDA and published in 2 separate publications some years ago. And we're looking at simulating the drug effect of around 3 milliseconds. And then if we use a highly precise ECG methodology, such as EPQT, we need 9 subjects in active and 6 on placebo to exclude that the effect has a larger effect than 10 milliseconds, to ensure that the upper bound is below the 10 milliseconds. If we use the same assumptions, so small underlying effect, but with a method with a larger variability, 10 milliseconds, which is quite typical for a standard technique, we need roughly twice as many subjects and the same number of placebo subjects to demonstrate the same key objective, to demonstrate that we can exclude an effect above 10 milliseconds. Also, I think it's worth mentioning that sometimes when we conduct therapeutic studies, these are also today mostly performed with concentration QTc analysis. What used to be a standard sample size of between 44 to 60 subjects in the crossover design study is today 24 to 28 subjects. So a much, much more powerful way of measuring the QTc interval by using EPQT and concentration QTc analysis. The 2022 revision brought non-clinical assays into the decision process. And today we're talking about the 5.1 pathway, which applies to all drugs that can be given safely to healthy volunteers, and the 6.1 pathway, which applies to drugs that cannot be given to healthy subjects in a safe way, and therefore are studied in patients. These are mostly oncology compounds. When I say the current, this is taken from a publication by myself and Derek Leifman for Lilly, current meaning before 2022. If we used a first-in-human study, we were told that we would achieve concentrations, Cmax concentrations that exceeded Cmax steady state in patients by 2-fold. Whereas if you have a double negative non-clinical assays performed as per best practices, you only need to cover what is called the high clinical exposure in patients. So you only need to cover what you can see in patients. If you have a first-in-human study with a negative effect at these concentrations. The 6.1 pathway is clearly different. You perform a study without placebo, you look at the change from baseline instead of the placebo-corrected change from baseline, and if you can show that your drug doesn't cause anything about 10 milliseconds, you can claim you will have a label that says that this drug is unlikely to cause anything about 20 milliseconds. This can be approved by using standardized non-clinical assays as well. So this is one example, also published some years ago in collaboration with a sponsor. We're looking at a typical first-in-human study, and the left graph is a very typical scatter plot, and you can see there is so much data in the first-in-human study on the low side of concentrations, and not so much on the high end of concentrations. But even so, we have concentrations up to around 3,600 nanograms per mil. This is how the goodness of fit, this is how the concentration QTc relationship looks based on these data. So one analysis. And the red bars are there to demonstrate that the model, the model which is shown by the black line in the gray shaded area, captures the data in a good way. And we can see that the slope is somewhat positive, but we can also see that we can exclude an effect up to concentrations of around 3,600 nanograms per mil. Now, importantly, the high clinical exposure scenario, which is the worst case scenario in terms of PK in patients, is by those patients taking a 3A4 inhibitor. In those patients, you can estimate that Cmax at steady state is around 1,300 nanograms per mil, which is down here. So we have, in fact, shown that at concentrations almost threefold above the high clinical exposure scenario, we can exclude an effect about 10 milliseconds. In this scenario, there is no need to go back and look at non-clinical assays. This is a clearly negative assessment evaluation based on the first-in-human study, clearly, and will most likely lead to a clean label in terms of QTc effects. So the 5-point pathway applies to studies in healthy subjects. And these are 2 slides on just practical considerations. When you do this, make sure that you pair all the PK time points with ECG extractions. Make sure that the site is aware of this and that subjects are supinely resting in an undisturbed environment for at least 10 minutes. Typically, as Dr. Patel already has outlined, the SAD has more dose groups than MAD. And often higher doses / concentrations are achieved in SAD. But it makes sense to implement the procedures in both SAD and MAD, because in case you have substantial accumulation of either parent or metabolite, it may make sense to also analyze the MAD part. Make sure that you have a robust baseline, meaning that you should have at least, well, you should have 3 time points prior to dosing on day 1 in most examples. The primary endpoint in the QT evaluation is the change from baseline QTc, from which we model the placebo-corrected change from baseline using concentration QTc analysis. Baseline, as I said, can typically be conducted on day 1 prior to dosing, unless the drug has a substantial heart rate effect. Then it's important to capture QT-RR or ECG data also from drug-free conditions, and therefore we use day -1 drug-free baseline data for this purpose. So there is no need, also there is no other need than QTCF, so the standard correction factor, unless you expect a substantial increase of heart rate. In that case, you need something else. You need to capture a wider range of heart rate and calculate your correction factor on that. We do this by collecting day -1 ECG data. Some other sponsors do this or advocate using submaximal exercise or posterior maneuvers. In our view, there's little evidence to show that this is the same as a drug-induced increase of heart rate, but that's an ongoing discussion. So my conclusions based over these last few years is that early QT assessment in first-in-human trials is a full accepted way to confidently exclude a clinically relevant effect in the QTc interval and thereby waive the request for later therapeutic study. The revised S7B 14 guidance from 2022 will further encourage the use of first-in-human studies. It's really critically important to proactively liaise both with non-clinical laboratories to make sure that hERG and non-rodent in vivo assays meet best practices, but also, of course, with your CROs and with the core labs to make sure that procedures are in place. In my view, the therapeutic study will not completely disappear and will remain as a viable option in selected cases. But overall, combining EPQT with concentration QTC analysis, QT evaluation can be made much more efficient today as compared to only 10 years ago. And by that, thank you so much, and I would like to hand over to Dr. Kuder. Thank you.
Thank you, Dr. Darpo. It's a real pleasure to be part of this webinar and to have the opportunity to present information about the technology we have developed during these past couple of years, which have really used the most recent tools in artificial intelligence. And before going into this specific topic, I want to spend a few minutes talking about the QT technologies that was already highlighted by Dr. Darpo in his slides, which is called iCompass. iCompass is really the core technology for the EPQT services, and really this software was built on a very unique set of data from the International Long QT Syndrome Registry. And because we had access to these ECGs from these patients with the inherited form of the long QT, the software was exposed to a very large spectrum of T-wave format or T-wave shapes, and the software became extremely robust to good QT measurement despite these changes in T-wave morphology. So really this technology, as I said, is the core for the EPQT, but what we did, not only we developed the technology, but we also validated it in collaboration with the FDA. In 2011, we actually conducted a study that was based on data from 7 thorough QT studies in collaboration again with the FDA, and we demonstrated in this independent validation of the precision of the iCompass technology that we were at the top of the technology existing for measurement of QT interval. So following this software, you can have the best software, obviously, for doing QT. If the data you propose to the software is of bad quality, it's going to impact your outcome and your deliverables. So really at Clario, we continued pushing forward the development of cardiac technologies. And we created a team, and I want to acknowledge the team at this point, that we call the Advanced Computational Approaches to Cardiac Precision Science. And what is very unique in this team is that you have a set of experts in a very, again, large spectrum of expertise, from software engineers, data analysts, biomedical engineers, cardiologists, and cardiac electrophysiologists. And it's really with the combination of those expertise that we came up with a very novel approach for looking at the quality of ECGs. And maybe the first question is why? Why don't we just stick to the existing signal processing technologies that we have in our toolbox? And really what happened is that with the rise of artificial intelligence and machine learning, for us as engineers and inventors, we really have in our hands now a really new set of tools that really transform our regular linear processing blocks that we have been using for decades in new tools that are now nonlinear equivalent, and that actually have the ability to create tools that have very unique set of performances. So what we've done really in this work is using these AI ML tools to assess the quality of the signal early in the analysis of data, before we do any kind of QT measurement or any kind of signal analysis. So what type of artificial intelligence and machine learning we use. And as you see here on the schema, you have actually the different categories. And when you do that with ECGs and electrocardiographic information, you can either extract information from the ECG, such as interval, amplitude of wave, and feed a model with this data, or with the most recent and most interesting tools of this category, using neural networks and deep learning, you can actually directly feed the model with the ECG signal, where there's no actually extraction of any information, but just feeding again the model with the signal itself. It's a very powerful way of looking at creating this model, but it has a constraint, and the constraint is really that such type of model, the design of this model requires a lot of data, okay? So we have a new tool, but we have also at Clario access to millions of de-identified ECG. So we had an opportunity here to really combine these two advantages and come up with some kind of new model for assessing the quality of ECGs. Before describing to you a little bit more the method, I want to highlight that there is another advantages to develop these machine learning technologies, is that once they are designed, it's not the end of it. It's really only the beginning. And really these tools are over time improved and are fed with new data. So we have in place not only a mechanism to develop new tools, but we also have a mechanisms to improve those models over time. It's a limitless, actually, ability to improve the technology. However, the way we do that has to be done under a certain, what we call good machine learning practices. And we are following at Clario what is recommended by the FDA for what is called software as a medical device technologies, where the FDA imposes or recommend, I should say rather, a whole framework on how to design the way these models are improved over time. So let's go into our model, and let me be a little bit more technical in terms of describing what we have done here. So the objective was really to have a method that gives us for every bit of the ECG an assessment, a quantitative assessment of the quality of the signal, okay? So what we've done, we have actually created a model that takes into input what we call an ECG epoch. An ECG epoch is a 3.6 second interval that is centered on a specific bit. We feed then to the model, and the model is designed to deliver a score that is going to vary between 0 and 1, where 0 represents a poor quality signal and 1 represents a good quality signal. We basically use the information we had at Clario, which were these millions of ECG that have been reviewed by humans and ECG tech and cardiologists, and that had labels this bit either good or what we call poor. We did that following the 3-bit on lead to methodology. But really what we had in our hands is really millions of bits that were reviewed and that had a label of good or poor. And we could use this data to create a model that would be actually predicting the quality of the signal based on this reference data. Please bear with me with this presentation, which is a little bit technical, but let me try to clarify it. At the top, you have actually a way of showing that this model is going to compute the epoch score, the quality score for every bit. So the epoch is just translated on the ECG lead for the whole recording, and that can vary from just an ECG strip to a 24-hour Holter signal. And really the method, the model will give you for every bit one of these score, again, that varies between 0 and 1. What I plotted on the bottom of this slide is what happened when we have all the score for a given time point. And at the top, you have this long colored, I would say, line that represent actually the quality of the overall time point using a color scale where the green represents a high value of the quality score, meaning that your ECG or the bit is actually of good quality. And when you have a color that is going towards hot color red or orange, it means that the quality of the score is actually low and you have poor bits. A little bit below, you have a presentation of the ECG strip and the strip for the same score. And you see clearly that with this kind of system, we can very easily have an idea of what is the quality of every bit on an ECG. So how did we develop this model? We use a set of 350,000 ECGs that were extracted from 118 studies. The way you develop this machine model, this machine learning ML model, is in three steps, okay? The first one is the training, where you develop the raw model. And here in this study, in this project, we use 80 studies in which there were 929 participants and represented a little bit more than 80,000 epochs. We designed the model, and we have a first step that we call validation, which is really tuning of the model, where we use another set of studies that is common with the training. So it's a 74 studies where all the studies were not different from the training set, but there were additional ones. And in the validation set, we had 753 subjects from 74 studies representing a little bit less than 12,000 ECGs. So at the end of the validation, we have a model that we are very confident with, and we use this model to do the final step, which is the test set, where now we're going to test the model on completely independent set of ECGs. And we had an additional 38 studies, including 1,210 participants and 257,000 ECGs. So roughly, we use 24% of the whole data for training, 3% for the validation, and 74% for testing. So this slide shows you the results, and let me explain a little bit the results. So we had actually for each ECG the label of poor or bad done by cardiologists. And what we've done, we use basically this validation or test set for validating the model, and we look whether the model tells us the good label for the ECG and for every ECG epochs. And these are the results, okay? So true positive, which is at the right lower part, is showing a 97% true positive. What does it mean? It means that when AI detected that an epoch, an ECG epoch was good, it was actually good. The true negative was 93%. So basically what it means is that when the model actually found out that the epoch was of poor quality, it was indeed of poor quality in the data set. So again, when we use the 3-bit only 2 methodology and labeling every bit, we basically have a model now that is extremely performant as identifying which epochs are poor or good quality. Now we push this concept a little bit further. Can we use now these quality metrics to know if one can extract at least 3 validate ECG replicates in every time point? Okay, so basically we have at this point the score on the data that is computed extremely quickly now when the data is submitted, and we have the ability to accumulate the score for each epoch for an overall time point and for every strip that is extracted from the time point. And we just did a very simple, I would say, a rule where we said, okay, if you have a cumulative quality score above such threshold, can we actually say that we have access to at least 3 good quality replicates per time point, okay? And basically, this table tells you the results of this, and these results were amazing, almost magical, because true negative were 100% and false positive were 0%. And again, these are in 3 independent data sets from the training, the validation that we have shown before. It was just new data from 2 SAT study and from a MAT study, including 58, 56, and 56 subjects. And what it tells you is that we have 100% specificity for detecting time points that do not generate sufficient usable replicates, okay? I wanna make sure it is understood. It means that we have a model that even we have anybody looking at the ECG can tell you with 100% specificity that the data can be used or not be used actually for extraction of the replicate from the Holter ECG. And not only that, but the model did not produce false positive results by labeling time point as usable that would be in truth to be unusable for analysis. So there's no error where basically bad data wouldn't be actually detected. So this is a very interesting result, and it is actually a very unique tool that at this point I've never seen done by anybody. How do we use this technology again to service our sponsors? And we have at Clario the collect, clean, and store service where you can send your data. And with this technology now, we can at very early stage of submission of your data, be able to assess whether the quality is good enough for doing your analysis, your EPQT analysis, and your QT interval extraction. So I'm going to summarize here the points is just to say that we developed an AI application to assess the quality of continuous ECG tracing collected in early phase clinical trials. We leverage this new technology in early phase studies to assess quality, data quality in near real time. This tool can quickly confirm if the data is analyzable and complete in lower dose cohorts. If required, correction can be implemented to ensure optimal data quality in subsequent higher dose cohorts. The AI quality tool can also perform a review of the data from a collect, clean, and store study, as I just said, providing assurance to the sponsor that store data will be of sufficient quality to support a future analysis. And this is only the beginning. We have, at this point, multiple projects ongoing that are going to leverage machine learning, artificial intelligence, in order to create a whole new landscape of tools and processes that will increase the confidence in all the analytics that are delivered on the data presented to the company. So we will enhance services, management of data, review of data in a way that has not been done so far. So I'm going to stop here, and I would like to give the lead to Dr. Patel for the Q&As. Thank you.
Thank you very much, Dr. Kader. So that concludes the presentation part of the webinar. At this point, please open up your chat and your question box, and please answer any questions that you may have for the panel. I'll take a look and see if we have anything coming through. Yeah, thank you. There are a couple of questions coming through, and here's one that I think actually would be best answered by Dr. Darbo. So the question is, since the high clinical exposure scenario is not known when you're conducting the first-in-human study, does it really make sense to include ECG monitoring matched with PK time points? And if so, is it better to do in the SAD part or the MAD part of the study?
It makes sense because typically the SAD part of a first human trial achieves concentrations that will not be seen later in development. And more often the SAD part has higher concentrations for both parent and metabolized, by the way, than the MAD part. In some cases where you see substantial accumulation, either of parent or analyte, it may also make sense to analyze the MAD part. But the principle, I think, or a good principle, is to make sure that you implement procedures in both parts. Both sponsors would then clearly analyze the SAD component and look at the PK to determine whether the MAD component should be analyzed. It comes with several advantages. One being, of course, that you would be aware of a potential QTc effect. The other one being that the SAD part captures sufficiently high concentrations to ensure that you can at least potentially waive the request for a therapeutic study later in development.
Thank you, Dr. Darpo. That was very clear and direct to the point and very helpful. There's another question here that I'm seeing. This is probably appropriate for Dr. Kuder to answer with a question about AI. So, Dr. Kuder, the question is, what are the next steps in the development of AI-powered ECG tools?
Thank you for this question. So there are many things happening around the development of technologies. And really, as I said in the conclusion of my presentation, those tools give us a new set of way of really measuring various aspects of ECG signal. And there are a lot of tools that can be used to identify not only the quality, but changes over time. What's happening to an ECG in the same subject across a study that can last for a long period of time, and very quickly identify what are the type of changes that happen to the signal. It could be different from simply QT interval prolongation due to drug, and there are many tools that we are working on to really, I would say, enhance the serial analysis of ECGs over time.
Thank you, Dr. Kuder. We're getting close. Maybe we have time for one more question. And the next one that's coming up is probably best for myself to answer. So the question is, what are the most common cardiotoxic adverse events that lead to termination of a drug development program? And so typically what we've seen, and there is literature on this, is that this is obviously dependent on the class of drug, but the most common adverse event that really stops drug development is heart failure. This happens more in oncologic assets as expected, where there's more myocardial toxicity, but you can see this up to 11 to 20% of the time in early phase drug development. And then the next most common cause for why an asset may be stopped in development is because of QTc prolongation, and that happens about 5 to 10% of the time. Again, based on the asset and therapeutic area in neuroscience and psychiatry, where you're looking at assets that are targeting the neuronal conduction, they also have off-target effects on the heart. So that's what we're seeing most commonly that will stop your program. This is important to keep in mind so you can try to understand these and mitigate those going forward as we can. So I think at this point, we're near the conclusion of our time, and I'd like to turn the presentation back over to Sonia so she can give us the wrap-up and concluding remarks. Sonia?
Okay, thank you very much, Dr. Patel, Dr. Darpo, and Dr. Kuder for answering all those questions, and of course for your great presentation. And just as Dr. Patel said, we have come to the end of the Q&A portion of the webinar. So if we could not attend to your questions, be rest assured that the team at Clario will follow up with you after this presentation. Now, for those of you interested in hearing more from Clario, and who would it be, you can register for the next webinar, Maximizing Value in Early Phase Oncology Trials, Innovative Endpoints for Early Insights, scheduled for Wednesday, February the 25th, and I hope that you'll be able to join us, and I put that link in the chat box to make it easy for you to register. Okay, let's go to the next slide here. Now, if you have any further questions, please direct them to the email address that you see there on your screen, and it's science@clario.com. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be pumping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Additionally, I have shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues once they register for the recording here as well. And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to such features as viewing the recording and more. Now, please join us in thanking our speakers for their time here today. So thank you again, Dr. Patel, for being my co-moderator, Dr. Darpo, and Dr. Kuder. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Bye.
Bye now.
Bye.